Inbreeding is the sexual reproduction of offspring from the mating or breeding of individuals or organisms that are closely related genetically. By analogy, the term is used in human reproduction, but more commonly refers to the genetic disorders and other consequences that arise from incestuous sexual relationships and consanguinity. Inbreeding results in homozygosity, which can increase the chances of offspring being affected by recessive or deleterious traits. This generally leads to a decreased biological fitness of the population, which is its ability to survive and reproduce. An individual who inherits such deleterious traits is referred to as inbred. The avoidance of such deleterious recessive alleles caused by inbreeding, via inbreeding avoidance mechanisms, is the main selective reason for outcrossing. Crossbreeding between populations also often has positive effects on fitness-related traits. Inbreeding is a technique used in selective breeding. In livestock breeding, breeders may use inbreeding when, for example, trying to establish a new and desirable trait in the stock, but will need to watch for undesirable characteristics in offspring, which can then be eliminated through further selective breeding or culling. Inbreeding is used to reveal deleterious recessive alleles, which can then be eliminated through assortative breeding or through culling. In plant breeding, inbred lines are used as stocks for the creation of hybrid lines to make use of the effects of heterosis. Inbreeding in plants also occurs naturally in the form of self-pollination. Overview. Offspring of biologically related persons are subject to the possible effect of inbreeding, such as congenital birth defects. The chances of such disorders is increased the closer the relationship of the biological parents. This is because such pairings have a 25% probability of producing homozygous zygotes in the offspring, containing two recessive alleles, which produce disorders. Because most recessive alleles are rare in populations, it is unlikely that two unrelated marriage partners will both be carriers of the alleles. However, because close relatives share a large fraction of their alleles, the probability that any such deleterious allele is inherited from the common ancestor through both parents is increased dramatically. It should also be noted that, in terms of probability, for each homozygous recessive individual formed, there is an equal chance of producing a homozygous dominant individual, one completely devoid of the harmful allele. Contrary to common belief, inbreeding does not in itself alter allele frequencies, but rather increases the relative proportion of homozygotes to heterozygotes. However, because the increased proportion of deleterious homozygotes exposes the allele to natural selection, in the long run its frequency decreases more rapidly in inbred population. In the short term, incestuous reproduction is expected to produce increases in spontaneous abortions of zygotes, perinatal deaths, and postnatal offspring with birth defects. The advantages of inbreeding may be the result of a tendency to preserve the structures of alleles interacting at different loci that have been adapted together by a common selective history. Malformations or harmful traits can stay within a population due to a high homozygosity rate, and it will cause a population to become fixed for certain traits, like having too many bones in an area, like the vertebral column in wolves on Isle Royale, or having cranial abnormalities in northern elephant seals, where their cranial bone length in the lower mandibular tooth row has changed. Having a high homozygosity rate is bad for a population because it will unmask recessive deleterious alleles generated by mutations, reduce heterozygote advantage, and it is detrimental to the survival of small, endangered animal populations. When there are deleterious recessive alleles in a population, it can cause inbreeding depression. The authors think that it is possible that the severity of inbreeding depression can be diminished if natural selection can purge such alleles from populations during inbreeding. If inbreeding depression can be diminished by natural selection, then some traits, harmful or not, can be reduced and change the future outlook on a small, endangered populations. There may also be other deleterious effects besides those caused by recessive diseases. Thus, similar immune systems may be more vulnerable to infectious diseases. Inbreeding history of the population should also be considered when discussing the variation in the severity of inbreeding depression between and within species. With persistent inbreeding, there is evidence that shows inbreeding depression becoming less severe. This is associated with the unmasking and eliminating of severely deleterious recessive alleles. It is not likely, though, that eliminating can be so complete that inbreeding depression is only a temporary phenomenon. Eliminating slightly deleterious mutations through inbreeding under moderate selection is not as effective. Fixation of alleles most likely occurs through Muller's ratchet. When an asexual population's genomes accumulate deleterious mutations that are irreversible. Genetic disorders. Autosomal recessive disorders occur in individuals who have 2 copies of the gene for a particular recessive genetic mutation, except in certain rare circumstances, such as new mutations or uniparental disomy. Both parents of an individual with such a disorder will be carriers of the gene. These carriers do not display any signs of the mutation and may be unaware that they carry the mutated gene. Since relatives share a higher proportion of their genes than do unrelated people, it is more likely that related parents will both be carriers of the same recessive gene, and therefore their children are at a higher risk of a genetic disorder. The extent to which the risk increases depends on the degree of genetic relationship between the parents. The risk is greater when the parents are close relatives and lower for relationships between more distant relatives, such as second cousins, though still greater than for the general population. A study has provided the evidence for inbreeding depression on cognitive abilities among children, with high frequency of developmental disability among offspring in proportion to their increasing inbreeding coefficients. Children of parent-child or sibling-sibling unions are at increased risk compared to cousin-cousin unions. Inbreeding may result in a far higher phenotypic expression of deleterious recessive genes within a population than would normally be expected. As a result, first-generation inbred individuals are more likely to show physical and health defects, including reduced fertility both in litter size and sperm viability, increased genetic disorders, fluctuating facial asymmetry, lower birth rate, higher infant mortality, depression on growth rate, smaller adult size, loss of immune system function. It can also occur in a large population if individuals tend to mate with their relatives, instead of mating at random. Many individuals in the first generation of inbreeding will never live to reproduce. Over time, with isolation, such as a population bottleneck caused by purposeful breeding or natural environmental factors, the deleterious inherited traits are culled. Island species are often very inbred, as their isolation from the larger group on a mainland allows natural selection to work on their population. This type of isolation may result in the formation of race or even speciation, as the inbreeding first removes many deleterious genes, and permits the expression of genes that allow a population to adapt to an ecosystem. As the adaptation becomes more pronounced, the new species or race radiates from its entrance into the new space, or dies out if it cannot adapt and, most importantly, reproduce. The reduced genetic diversity, for example due to a bottleneck, will unavoidably increase inbreeding for the entire population. This may mean that a species may not be able to adapt to changes in environmental conditions. Each individual will have similar immune systems, as immune systems are genetically based. When a species becomes endangered, the population may fall below a minimum whereby the forced interbreeding between the remaining animals will result in extinction. Natural breedings include inbreeding by necessity, and most animals only migrate when necessary. In many cases, the closest available mate is a mother, sister, grandmother, father, brother, or grandfather. In all cases, the environment presents stresses to remove from the population those individuals who cannot survive because of illness. There was an assumption that wild populations do not inbreed. This is not what is observed in some cases in the wild. However, in species such as horses, animals in wild or feral conditions often drive off the young of both sexes, thought to be a mechanism by which the species instinctively avoids some of the genetic consequences of inbreeding. In general, many mammal species, including humanity's closest primate relatives, avoid close inbreeding possibly due to the deleterious effects. Examples. Although there are several examples of inbred populations of wild animals, the negative consequences of this inbreeding are poorly documented. In the South American sea lion, there was concern that recent population crashes would reduce genetic diversity. Historical analysis indicated that a population expansion from just 2 matrilineal lines were responsible for most individuals within the population. Even so, the diversity within the lines allowed great variation in the gene pool that may help to protect the South American sea lion from extinction. In lions, prides are often followed by related males in bachelor groups. When the dominant male is killed or driven off by one of these bachelors, a father may be replaced by his son. There is no mechanism for preventing inbreeding or to ensure outcrossing. In the prides, most lionesses are related to one another. If there is more than one dominant male, the group of alpha males are usually related. 2 lines are then being line-bred. Also, in some populations, such as the crater lions, it is known that a population bottleneck has occurred. Researchers found far greater genetic heterozygosity than expected. In fact, predators are known for a low genetic variance, along with most of the top portion of the trophic levels of an ecosystem. Additionally, the alpha males of 2 neighboring prides can potentially be from the same litter. One brother may come to acquire leadership over another's pride, and subsequently mate with his nieces or cousins. However, killing another male's cubs, upon the takeover, allows the new selected gene complement of the incoming alpha male to prevail over the previous male. There are genetic assays being scheduled for lions to determine their genetic diversity. The preliminary studies show results inconsistent with the outcrossing paradigm based on individual environments of the studied groups. In Central California, sea otters were thought to have been driven to extinction due to overhunting, until a colony of about 30 breeding pairs was discovered in the Big Sur region in the 1930s. Since then, the population has grown and spread along the central Californian coast to around 2,000 individuals, a level that has remained stable for over a decade. Population growth is limited by the fact that all Californian sea otters are descended from the isolated colony, resulting in inbreeding. Cheetahs are another example of inbreeding. Thousands of years ago, the cheetah went through a population bottleneck that reduced its population dramatically, so the animals that are alive today are all related to one another. A consequence from inbreeding for this species has been high juvenile mortality, low fecundity, and poor breeding success. In a study on an island population of song sparrows, individuals that were inbred showed significantly lower survival rates than outbred individuals during a severe winter weather-related population crash. These studies show that inbreeding, depression, and ecological factors have an influence on survival. Measures of inbreeding. A measure of inbreeding of an individual A is the probability F that both alleles in one locus are derived from the same gene in an ancestor. 2 alleles derived from the same gene in an ancestor are said to be identical by descent. This probability F is called the coefficient of inbreeding. Another useful measure that describes the extent to which 2 individuals are relatives is their co-ancestry coefficient F, which gives the probability that, taking one random allele from A and another random allele from B, both are identical by descent. This is also denoted kinship coefficient between A and B. A particular case is the self-co-ancestry of individual A with itself, F, which is the probability that taking one random allele from A and then, independently and with replacement, another random allele also from A, both are identical by descent. Since they can be identical by descent by sampling the same allele or by sampling both alleles that happen to be identical by descent, we have F equals 1/2 plus F2. Both the inbreeding and the co-ancestry coefficients can be defined for specific individuals or as average population values. They can be computed from genealogies or estimated from the population size and its breeding properties, but all methods assume no selection or are limited to neutral alleles. There are several methods to compute this percentage. The two main ways are the path method and the tabular method. Typical co-ancestries between relatives are as follows: father, daughter, mother, son or brother, sister 25%. Grandfather, granddaughter or grandmother, grandson 12.5%, half-brother, half-sister, double cousins 12.5%, uncle, niece or aunt, nephew 12.5%, great-grandfather, great-granddaughter or great-grandmother, great-grandson 6.25%, half-uncle, niece or half-aunt, nephew 6.25%. First cousin 6.25%. Domestic animals. Breeding in domestic animals is primarily assortative breeding. Without the sorting of individuals by trait, a breed could not be established, nor could poor genetic material be removed. Homozygosity is the case where similar or identical alleles combine to express a trait that is not otherwise expressed. Inbreeding, through homozygosity, exposes recessive alleles. Breeders must avoid breeding from individuals that demonstrate either homozygosity or heterozygosity for genetic-based diseases. The goal of preventing the transfer of deleterious genes may be achieved by reproductive isolation, sterilization, or, in the extreme case, culling. Culling is not strictly necessary if genetics are the only issue in hand. Small animals such as cats and dogs may be sterilized, but in the case of large agricultural animals, such as cattle, culling is usually the only economic option. The issue of casual breeders who inbreed irresponsibly is discussed in the following quotation on cattle. Meanwhile, milk production per cow per lactation increased from 17,444 pounds to 25,013 pounds from 1978 to 1998 for the Holstein breed. Mean breeding values for milk of Holstein cows increased by 4,829 pounds during this period. High-producing cows are increasingly difficult to breed and are subject to higher health costs than cows of lower genetic merit for production. Intensive selection for higher yield has increased relationships among animals with inbreed and increased the rate of casual inbreeding. Many of the traits that affect profitability in crosses of modern dairy breeds have not been studied in designed experiments. Indeed, all crossbreeding research involving North American breeds and strains is very dated if it exists at all. The BBC produced two documentaries on dog inbreeding titled Pedigree Dogs Exposed and Pedigree Dogs Exposed. 3 years on that document the negative health consequences of excessive inbreeding. Line breeding is a form of inbreeding. This method can be used to increase a particular animal's contribution to the population, while line breeding is less likely to cause problems in the first generation than does inbreeding. Over time, line breeding can reduce the genetic diversity of a population and cause problems related to a too small genopool that may include an increased prevalence of genetic disorders and inbreeding depression. Outcrossing is where two unrelated individuals are crossed to produce progeny. In outcrossing, unless there is verifiable genetic information, one may find that all individuals are distantly related to an ancient progenitor. If the trait carries throughout a population, all individuals can have this trait. This is called the founder effect. In the well-established breeds that are commonly bred, a large gene pool is present. For example, in 2004, over 18,000 Persian cats were registered. A possibility exists for a complete outcross, if no barriers exist between the individuals to breed. However, it is not always the case, and a form of distant line breeding occurs. Again, it is up to the assortative breeder to know what sort of traits, both positive and negative, exist within the diversity of one breeding. This diversity of genetic expression, within even close relatives, increases the variability and diversity of viable stock. Laboratory animals. Systematic inbreeding and maintenance of inbred strains of laboratory mice and rats is of great importance for biomedical research. The inbreeding guarantees a consistent and uniform animal model for experimental purposes and enables genetic studies in congenic and knockout animals. The use of inbred strains is also important for genetic studies in animal models, for example to distinguish genetic from environmental effects. The mice that are inbred typically show considerably lower survival rates.